Well, thank you, and good morning to everyone. Thank you, Nadine and Julia, for the introduction, and certainly for working with you and the society all these years. Good morning, everyone. I'm a human geneticist by training, so neither an engineer, nor a clinician, nor a microfluidics expert. So it's just been a pure joy and pleasure to be part of the MPS group from the beginning about 10, 11 years ago. So in my presentation today, I'll probably tell you a little bit more about NCATS and the National Institutes of Health, why we're developing this technology, and then I'll give you some few selective examples of how MPS can be used in toxicology and pathology. So first up would be the NCATS, or the National Center for Advancing Translational Sciences. This is probably the newest institute within the NIH family of centers and institute. We were established by the United States Congress back in 2012, primarily with a mission to advance the development of innovative tools and technologies for diagnostics and therapeutics across many diseases and conditions. So the question would be, why does even United States Congress care about this area of developing tools and technologies? Where really they have tasked us to look into what's going on or what's broken with the drug development pipeline. And so shown here are an example or an illustration of where things are needed in MPS. I think you've heard in the course of this meeting of the MPS World Summit that it's a relatively inefficient process. Anything that succeeds only 10% of the time in my book or anyone's book is really, that high attrition rate doesn't show a lot of efficiency. A good number of failure comes from lack of efficacy, about 55% to 60%, and about 28 to 30% is primarily from toxic effect or safety profile. And if you look into why that is failing, there's quite a wide range in terms of the indication. If you look at in hematology, they tend to do a little bit better in developing drugs for hematologic indications, about 26%. But if you look at the lower end of the spectrum for oncology drugs, that's at 5%. And so you get sort of like an average of about 9.6%. And the reason for that is the lack of concordance of toxicities across many different conditions, comparing human toxicity with animals. So you see that this in some ways mirror this, where you tend to do well in certain areas, organ systems, but not necessarily in most of them. And you've heard it also that it takes a long time to develop drugs, 10 to 15 years at a cost of $2.6 billion. And so what we really need are really novel tools that would be more predictive of human response. And that's where we come in. So here's some additional facts to consider. Over half of all the drugs that gain FDA approval actually gets withdrawn. And the sad thing is adverse drug reactions is actually the fourth leading cause of death in the United States. About 100,000 patients annually at a cost of about $4 billion. So that's even in addition to the cost of development of drugs. And then more than 2 million Americans receive emergency hospital treatment. And this is just in a snapshot in 1 year because of adverse reaction to prescribed medication. And so this is sort of like the toxicology dilemma. You now have research in animals, whatever species they are. And the central question, of course, is how do you get to a phase 1 trial where you have primarily healthy volunteers and expecting animals to reflect physiological conditions of people. And then, of course, the way animal studies are conducted is also quite limiting in the sense that also have limited number of animals that can be tested, and that's primarily because of animal concerns as well. And then there's going to be species and strain differences, and then animal models are also genetically and physiologically distinct from each other. So you'll find that sometimes my rat data is not concordant or discordant with dog data. And so they sometimes don't agree with each other as well. Of course, there's age, gender, and health status differences. Most animals are under controlled exposures, whether it be diet or behavior. And then, of course, there's the ethical animal concerns. And so now we have the, what we call the parallelogram model of toxicology, in the sense that most commonly used model systems assume that there are structural homologies, identical target, as well as distribution, signal transduction pathways across many different species. And of course, we know that that's not true. And so that brings then down to human relevance. Are the human toxicities identified in phase 1, they're not always going to be predicted by the usual GLP preclinical species that we use, right? And so what are the right models that we need? Essentially models that are fit for purpose is what we need. So the fit for purpose means it mimics the human functional response. It should be translational to the clinic, and it should be physiologically relevant. That means functional change and structural changes should also be readily picked up, and of course has directly translatable and accessible pharmacodynamics as well as safety biomarkers. And this is not new. This is an OECD document that's been around since 2005, providing guidance in terms of how do you validate in vitro and in vivo tools. Essentially, it breaks down into 3 criteria. Number 1 is reliability. Reliability is essentially a measure of the extent that an assay or a test or a model can be performed reproducibly, and that's a key word, within and between laboratories over time. The next criteria would be relevance. This is essentially where the fit for purpose comes in, where it describes the relationship of the test to the interest that you want. Oops, sorry. Whether it's meaningful and useful for a particular purpose. And then the last criteria is, of course, regulatory acceptance, which is essentially the formal acceptance of a test by regulatory authorities, indicating that the test method and the data gathered by that test method actually meets the specifications of regulatory agencies. Now, to give you an illustration about MPS, so in this particular case, we're talking about the liver on a chip, or liver, to be more specific, a liver sinusoid on a chip. And why the liver? So when I spoke to pharmaceutical companies when we were beginning the MPS program, number 1 source of toxicity is, of course, the liver, which is not surprising because the liver is responsible for the concentration and metabolizing of majority of medications. And because of that, drug-induced liver injury is actually the most common cause of acute liver failure, about 15 to 20 per 100,000. And then adverse drug reactions are an important cause of liver injury. Of course, that eventually leads to withdrawal of the drugs. And so here's a representation of the liver sinusoid. This is actually from the Emulate platform. This is not intended to advertise for Emulate, but this is, again, just as an illustration, and I will use some examples of data coming out from the Emulate platform, but just more for illustration purposes, not necessarily an endorsement of their product. And so their liver on a chip will have essentially the hepatocytes, as well as parenchymal and non-parenchymal cells, as shown here. And now why is this relevant? So this is Emulate data that was published back in 2019, where they looked at a drug called pheoluridin. Pheoluridin is an antiviral agent that was developed for hepatitis B. It went through preclinical studies in rats and dogs, and obviously showed no safety signal, and so it went on to be approved for human clinical trials. They enrolled 15 patients, and 5 of those patients died, right? And so, of course, FDA had to withdraw it. So the question is why? And so here's an opportunity then to look at why that is. And so here's a comparison of human liver on a chip, and this is a rat liver on a chip. And then it was dosed with increasing doses of phenyluridine at 0, 1, 10, and 30 micromolars. And as apparent here with Nile red staining. Nile red staining would show yellow deposits if there's lipid deposits or an indication of steatosis. And so clearly in the rat, even in the rat liver on a chip, there was no indication of toxicity, whereas in the human liver on a chip, you can see a dose-dependent increase in steatosis, and that could be quantified on this chart right here on the right-hand side, showing again the dose-dependent increase. Of course, staining is always not necessarily the data that you would like. You would also want to see functionality, and the number one function of the liver that you look for in terms of function would be albumin secretion. If you look at albumin secretion in the rat liver on a chip, again, you see almost consistent albumin secretion without hardly any change, whereas in the human liver on a chip, you see a dose-dependent decrease in albumin secretion as well. And then looking for liver injury markers such as miR-122, glutathione S transferase, and keratin, again, you see a dose-dependent increase in the expression of liver injury markers. So that demonstrates not only pathology, both in terms of histological changes as well as also in the function of the liver. And then, of course, Emulate followed this up with a much more extensive study. Again, this shows how liver on a chip or liver MPS can be used to accurately predict hepatotoxicity. So this is now looking at a number of compounds. So these are looking at 22 known hepatotoxic compounds. So just like phenyluridine went through safety, preclinical safety assessment, successful, went to the trial, and was deemed to be safe based on animal data, but in the human trials caused more than 200 patient deaths and 10 liver transplants. So clearly toxic, withdrawn, and so then you're also looking at 5 known hepatotoxic, that is just to show that there's selectivity in the assay. And collectively, looking at all these, now what, 27 compounds, both hepatotoxins and non-hepatotoxic agents, human liver on a chip showed an 87% sensitivity and 100% specificity in being able to predict hepatotoxicity, certainly outperforming the liver spheroids, which is one of the most common preclinical assay for hepatotoxicity at 47%. Next example I'd like to show would be the importance of having a multi-organ system interconnected. So in this case, connecting liver and kidney. So in this particular case, let's talk about the kidney. So the kidney functions, of course, as we know, to eliminate about 20 to 25% of drugs in our system. Renal blood flow rate is about 20 to 25% of our cardiac output, so a tremendous amount of pressure, blood pressure going through the liver, and it accounts for reabsorption, water reabsorption that effectively concentrates luminal drugs and metabolites up to 100-fold. And then also 20% of all hospital admissions is because of acute kidney injury attributed to drug-induced kidney injury. So this is human kidney on a chip, probably more properly called proximal tubule on a kidney. This was developed by Jonathan Himmelfarb from University of Washington. This is a device manufactured by Nordis, and of course the illustration of where the proximal tubule of the kidney is. This particular kidney model has shown, just like the liver and the chip, to show robust, reproducible physiological kidney function. And what are those measures? That means they show polarity of the tubule and express key markers of proximal tubules such as aquaporin-1, GGT, and SGLT2 markers, as well as contains renal proximal tubule epithelial cells, and can live up to 24 days under flow. And again, the most common use is for indication would be using kidney injury marker or KIM1, and then using iPS-derived cells as well as a source for disease modeling. And so we've worked with Jonathan to model, for example, polycystic kidney disease from iPS-derived patient cells. And so here's an illustration of what an interconnected integration of multiple tissue types look like. So this is a collaboration within multiple groups and various scientific disciplines, from engineers, nephrologists, hepatologists, and things. So connecting the gut, the liver, the kidney, and then essentially connecting them by functional integration. So you have all different platforms here, but then it's like they're not necessarily physically integrated, but you take the flow through and pass it on to the next organ system. And here's the illustration of what the liver and the kidney integration would look like, especially looking at the mechanism of toxicity of aristolochic acid that causes nephropathy. So aristolochic acid is highly nephrotoxic as a class 1 carcinogen. It has been identified in Chinese herb medicine, but it's primarily used for weight loss. But unfortunately, it can cause nephropathy, and also been attributed here in Europe to the Balkan endemic nephropathy. Up until this time, the mode of action was not known, and the pathway of human bioactivation is not fully elucidated or not known. So if you look at staining of just the kidney by itself, treated with aristolochic acid at 0 micromolars or 10, no evidence of nephrotoxicity. But if you flow the aristolochic acid through the liver first and then to the kidney, again at 0, no nephrotoxicity, but then at 10 micromolars, the flow through from the liver shows kidney death, as shown here with KIM1 staining, and that's illustrated in this step as well. And so one of the things that was brought to light with this series of experiments is that aristolochic acid is broken down in the liver into a sulfated conjugated form, as shown here, and then you can actually block the entry of this conjugated sulfated form of aristolochic acid by probecenid, showing that you can block the nephrotoxicity, as shown here in this live dead cell staining. Another important point that I'd like to discuss would be how you can combine machine learning and AI in being able to, better go fast. And so here's work by Jamie Thompson's group at University of Wisconsin, actually, in Wisconsin, and Morgridge Institute. So his work is primarily looking and taking neural progenitor cells, shown here as NPCs, and then at day 9 incorporating endothelial cells and MSCs. And this is the type of staining that he gets from these cortical neurons. Here's more staining of different neuronal types from GABAergic, glutamatergic neurons, showing also astrocytes being present, oligodendrocytes and pericytes, But the important point I wanted to do is to point out how you can use AI and machine learning in MPS in being able to predict neurotoxicity. Sorry about that. I timed myself, so I have to rush. So here's a list of about 70 compounds that were tested that was used for training set. So about 39 of this, 1 to 39, are actually known neurotoxins, and the rest are control that are known to be non-neurotoxic. And so the brain constructs were treated with each of this compound. RNA data was collected, and signatures of those RNA transcriptome were then used to indicate whether the signatures show clear neurotoxicity or not. And so this is sort of like illustrated in this schematic where you have the brain construct here consisting of neurons, glia, microglia, and vasculature. Again, transcriptomic is the readout. This is more of a discovery. It's not a targeted transcriptomic, so looking at 19,000 possible transcripts, and then 60 training compounds, again indicating 34 were known to be neurotoxins, 26 control, and then once the system is trained, it was treated with 10 blinded compounds, 5 toxins, and 5 controls. Duplicate samples at least, so two replicates, and essentially shows that over the course of time, this algorithm using data coming out from the neurochip or neural construct showed 80% accuracy in being able to predict neurotoxicity, and for each time point, and overall 90% accuracy for the combined data set. So essentially, and then with the blinded set of compounds, meaning the 5 neurotoxins and 5 9 out of 10 was correctly predicted, and the one that was missed was a false positive. And this is published now in this paper. With that, I'll probably just rush through this. This is just showing efficacy, how you can use MPS also to look at disease models, be able to assess treatment, and this is also from others, so I'll skip to that and go to this summary slide in the sense of usefulness of MPS to study microbiome, toxin, infectious disease, various countermeasure agents, applying it to a number of different applications from personalized precision medicine to studies of diversity among populations, rare disease research, and of course drug efficacy and toxicity screening. And with that, I'll end and just show this accomplishment slide. Sorry for going over time.
So thank you very much. I think we have time for 1-2 questions depending on the length. So thanks for your talk. Any very burning question?
Thank you very much for your talk. So I may have a very naive question, but we've seen a few talks this morning talking about patient stratification, in which drug toxicity and efficacy could vary between patients. So the question is, could the result that we achieve with MPS be biased by the use of a single cell line or a cell line derived by a single patient, rather than using an array of cells with different genetic makeups?
No, so really it's when you talk about patient stratification, like in the session this morning on precision medicine, that is looking actually at representation of various patient cells, not just a single cell line. So that's how you can stratify. You represent essentially the patient demographics on the chips and be able to identify who are likely to suffer from adverse event, who are likely to benefit from the treatment. And so then in some ways you can actually see almost like a 3 Rs being applied in the clinical study. So you don't necessarily have to have, let's say, 10,000 individuals enrolled in a phase 3 clinical trials, because you would have stratified the population in terms of who are most likely to respond to the treatment based on either the mutation or based on their pharmacogenomic profile. That's what we call about patient stratification. So, but it's not based on just a single cell line.
Yeah, thank you,
I
Dan.
know it.
Yeah, very good. I just want to emphasize the importance of standardization, especially of terminology. You mentioned very good point about the kidney. Although we read papers say kidney on a chip, but that doesn't exist. So what exists is mostly proximal tubule on a chip or glomerulus on a chip.
Right.
So yeah.
Yeah, so that's why at NIH we consciously call them tissues on chips instead of organs on chips. Because yeah, it is not an entire organ system. They're representation, and the idea behind that is you reconstruct the basic cytoarchitecture and the basic functional unit of an organ system, and typically that would just be a collection of cells and tissues. And then you can build complexity eventually, depending on your context of use. So you can, essentially it's building the chip, the MPS from the bottom up, depending on the biological question you would want to answer. Yeah. But that's a good point, yeah. We need to be cognizant of how we message all the time.